After presumptive screening has been performed on the DNA samples, such as blood, saliva, or semen, extraction can be conducted. Notably, samples that are suspected to have multiple contributors will undergo a specialized form of extraction called differential extraction. However, this topic will be discussed in greater detail in a following video. The main objective of DNA extraction is to recover the greatest quality and amount of DNA as possible. To do this, the samples must undergo two main steps of extraction: lysis and purification. Lysis refers to the breakdown of tissues and cells. The membrane of the cells within the sample must be disrupted to release the genetic material inside, the DNA. This can be done through the following: detergents, high temperatures, low levels of pH, and chemicals. Sodium dodecyl sulfate, or SDS, is a common detergent that is used, but will be discussed later in the video. Purification is the removal of proteins, inhibitors, cell debris, and any other unwanted molecules. Protein removal is important as nucleases, a type of enzyme, are known to degrade DNA. Proteinase K is a reagent that is frequently used for protein denaturation, which will inactivate the nucleases. There are three main methods used to extract DNA: organic extraction, Chelax, and solid phase extraction. As previously mentioned, Chelax is known for being economical and efficient as the sample and all the reagents are added into a single tube. Due to this, it was coined the dirty or quick and easy extraction. This extraction requires three main components: Chelax 100 resin, high temperatures, and incubation. The Chelax 100 resin binds and removes metal ions, magnesium in this case, that are required for nuclease activity. Prior to the addition of the Chelax solution, the sample is incubated in water at room temperature for 10 to 15 minutes. After the Chelax solution is added, the temperature is raised to 56 degrees Celsius, and the sample is incubated for another 30 minutes. A final incubation is performed at 100 degrees Celsius for 8 minutes. These incubation steps burst open the cell membrane, denature DNA or separate the two DNA strands, remove inhibitors and contaminants, and bind up magnesium ions. The tube undergoes centrifugation to enable the Chelax resin to form a pellet at the bottom of the tube. The remaining solution containing the DNA, known as the supernatant, is collected and transferred to a new tube. The supernatant can then be used to perform DNA quantification, or qPCR, and amplification, or PCR. Like Chelax, organic extraction requires centrifugation and incubation. But before performing these steps, 4 reagents are added to a tube containing the sample: SDS, dithiothiazole, or DTT, ethylene diamine tetraacetic acid, or EDTA, and proteinase K. As stated earlier, SDS is used to lyse the membrane of the cells while proteinase K removes proteins. EDTA is a chelating agent that prevents DNA degradation by inactivating nucleases, while DTT assists proteinase K with protein removal. The tube is then incubated at 56 degrees Celsius and centrifuged. After lysis, the DNA is then purified using 3 organic solvents: phenol, chloroform, and isoamyl alcohol, hence the name of organic extraction. These solvents separate and isolate the components within the sample to form 3 layers or phases: aqueous, interphase, and organic. It is important to consider the pH of the organic mix, as it can determine what molecules are found in each layer. If the organic mix has a pH between 7 to 8, DNA can be found in the aqueous layer, proteins in the interphase, and lipids and debris in the organic. On the other hand, if the organic mix is acidic, the composition of the layers will change. The aqueous layer will contain RNA as it is more polar than DNA. DNA would move to the interface as the acidic environment neutralizes it. Proteins and lipids would be found in the organic layer. Once the solvents are added, the tube will be vortexed and centrifuged once more to ensure distinct separation between the layers. Using a pipette, the aqueous layer, or the top layer, is transferred to a new tube with tris EDTA TE buffer. The sample can then undergo quantification and amplification to determine the amount of DNA that was recovered. Although organic extractions yield high amounts of DNA, the process requires a lot of time and effort. As a result, solid phase extractions are more popular. This method relies on the preferential binding of DNA to a silica membrane. Solid phase extractions can be categorized into two main types: spin columns and magnetic beads. These techniques are not only faster, but can also be automated, increasing throughput in forensic and research applications. Similar to organic extractions, the process begins with a lysis step, where the sample, in this case the cutting from the cotton swab, is treated with a lysis buffer containing detergents and proteinase K and incubated at 56 degrees Celsius. The cutting is transferred into a SpinX basket and centrifuged at maximum speed. Ethanol is added to the extract, vortexed, and briefly centrifuged. The resulting extract is then pipetted into a spin column using a silica-based membrane to isolate and purify the DNA. The purification process within the silica column consists of 4 key steps: DNA binding, washing, drying the silica membrane, and eluting the DNA from the membrane. DNA binding. This step is based on the affinity of DNA to silica when the salt concentrations and alcohol concentrations are high and the pH is low. High salt concentrations in the presence of alcohols such as ethanol disrupt the water structure and promote hydrogen bonding between the DNA and silica. The extract is pipetted into the column, where the DNA binds with the silica membrane inside the column. Proteins and other contaminants remain in solution. This is then centrifuged, which leads to the unbound molecules exiting the column, leaving the DNA and other loosely bound molecules behind. The next step is washing. 2 wash buffers are used to remove impurities that are loosely bound to the silica membrane. Wash buffer 1 and wash buffer 2 effectively wash away proteins, cellular debris, and other unwanted materials like salts that were initially present to facilitate the binding of DNA to the silica membrane. The silica membrane is then dried by centrifuging at high speed. The final step is eluting the DNA from the column. After the 2 wash steps and the drying step, the conditions that facilitated DNA binding are reversed, allowing the DNA to be released into the solution. The elution buffer is added to the column, allowed to incubate at room temperature, and centrifuged to get the DNA out of the column and into a collection tube. This DNA will then be quantified to determine the amount of DNA present in the extract obtained from the sample. Similar to the silica columns, magnetic beads are used in the isolation and purification of DNA. These are small magnetic beads coated with silica. Some forensic samples, like hair, nails, or bones, are termed challenging samples and require additional steps to extract DNA. These samples generally have lower quantities of DNA and have structurally different cells than those that are seen in traditional, more common forensic samples, such as blood, semen, and saliva. Therefore, DNA extraction from these types of samples requires longer incubation times, with agitation, and stronger detergents such as DTT in much larger quantities in the DNA lysis step. DNA is isolated from these extracts using solid phase extraction methods. In forensic labs, since DNA has to be extracted from a large number of samples, high throughput is a necessity. Automated machines with onboard liquid handling are used for the purification of lysates after sample lysis to increase throughput in forensic laboratories. These methods use solid phase extraction techniques, most commonly silica-coated magnetic beads for the isolation of DNA. An example is the EZ2 Connect, which can purify up to 24 sample lysates to isolate DNA using silica-coated magnetic beads. Now that DNA is extracted from the samples, quantification is carried out to assess how much DNA is present in the sample, which will be explained in detail in an upcoming video.